Vertebrates are, most of them are strict osmoregulators. We call them osmoregulators. They regulate their water and solute concentration. Most of them maintain their intracellular and extracellular fluids that are somewhere between 200 and 400 milliosmoles, right? So on your test, I just picked 300, right? 300 is kind of in the middle. Most terrestrial organisms like us that live on land maintain an osmolarity of about 300 milliosmoles internally, no matter what's going on around them. And so if you start thinking about fish in particular, animals that live in water, it's pretty incredible what they do to maintain those types of osmolarity numbers internally. So freshwater salamanders like this guy here, that's the axolotl, or freshwater fish, they typically have internal osmolarities between 200 to 250 to 300 sometimes milliosmoles, where freshwater is 50 milliosmoles as far as salt goes. So it's very hypoosmotic, right? That is, there's very little salt, lots of water. And so they're going to tend to want to take water up, right? Water's going to try to get into their bodies, and they're going to lose salt because salt wants to move, even though it has a hard time doing it, right? It's going to be a much higher concentration inside the animal than outside. And where they gain all their water is over their gills, mostly, because in this animal, the gills are these little frilly, looks like a mane around them, right? They're called external gills. This is where they exchange oxygen for carbon dioxide. I picked this salamander here because it's not a true albino, because you can still see it has some eye color. But it is it is very light, and you can see the gills are red because they're not pigmented, and that's blood that's running through them. That's why they're really bright red. And so oxygen goes in, carbon dioxide goes out. Well, if you can move those two gases, you're going to move water also. I mean, it's just really hard to stop water from moving. And so they take up a lot of water into their gills, and then they have to get rid of that water so that it doesn't dilute their salts. And they do that using their kidneys. All right, so the kidneys are going to be the main player here that gets rid of water. And we're going to talk about the glomerulus again here coming up, and the Bowman's capsule. And they have absolutely humongous Bowman's capsules, or in glomeruli, which is the first part of the nephron in their kidney that filters blood, because they need to filter lots of that. They just need to filter water. And they're basically urinating all day long, a very dilute urine. Their kidneys are pretty impermeable to salt, but it's almost impossible not to lose some salt too, right? No matter how well the kidney is at preventing salt loss, they are going to lose some salt to their urine. And because of that, even if it's a little bit, over time, that's going to dilute, right, their body fluids. So they have to pump salt, which is the phenomenal thing, from water that has no salt. To do that, they're going to use the gill epithelium, and these cells are going to be called chloride cells, and they have the ability to suck salt out of fresh water, and we're going to talk about that. Either in the gills in fish, or the skin is what really does that in amphibians. In turtles, freshwater turtles, it's the bladder that can pump salts and remove salts from freshwater. And then the kidneys in mammals. What's really cool are all the cells that line these very different organs that are responsible for this do it the same way, or in a very similar way. So now the integument is impermeable to water, the gills gain water over, and the kidneys are going to filter all that water out and lose some salts. Their skin, in the amphibians at least, is going to be responsible for pumping salt, and their gills will do this in the fish. So now marine animals, you can do osmolarity problems in two ways. You can be a conformer or a regulator. As I've already mentioned, most vertebrates are regulators, right? There are a few exceptions. When we look at the vertebrate group, Hagfish are probably the most primitive as far as the way that they deal with osmolarity. They do it like invertebrates do. Most marine invertebrates and your primitive chordates, your protochordates like Amphioxus, they are osmoconformers. They are isotonic to saltwater. They typically have the same osmolarity as saltwater. And saltwater is about 1,000 milliosmoles. All right, anywhere from 1,000 to 1,200 milliosmoles. So it's really hypertonic. And most of these organisms concentrate sodium chloride as their main inorganic salt. All right, these ions here, almost every organism animal stays away from. We don't want to concentrate calcium, magnesium, and this is phosphate, because calcium and magnesium are positive and sulfate is negative, and calcium and magnesium in particular play a major role in nerve and muscle physiology. So you don't want to mess with their concentrations, right? We want to have them at a certain concentration. We don't want to just let them flood the interstitial fluid. And so when we do concentrate salts, we're typically, if we're going to do salt to become a isotonic organism or an osmoconformer, we're going to concentrate sodium chloride. And that's what the hagfish does. So it will concentrate sodium chloride in its body tissues. Here's a hagfish right here. And mimic the osmolarity of saltwater. And that's why we do not include it in vertebrates, because all vertebrates are regulators. All right, all vertebrates are regulators, typically in one way or another. Now, I've always mentioned there are exceptions to the rule, right, of that regulation. Elasmobranchs, this word elasmobranchs, these are sharks and rays. It's the scientific name for sharks and rays. They do osmoconform, and they osmoconform not using salt, though. So they are strict regulators of any of the ions. They concentrate urea, which is a byproduct of amino acid breakdown. Okay? So urea is a small organic compound that carries two ammonia groups, all right, which are, if you remember, you guys all remember what amino acid looks like? When you break down amino acids, let's get a piece of chalk here. Your amino acid, if you remember, has a carbon. An alpha carbon has a hydrogen, and it has a carboxyl group and a amino group, right? And then this is an R group. So if we remove this, we can use this compound to do other things, right? Like we could break it down for sugar, or we could treat it like a carbohydrate and use it to make ATP. But this right here, ammonia NH3, or ammonium ions, NH4, they are toxic. I mean, really, really toxic. All right, so they raise pH and can wreak havoc across interstitial fluid. And so we need to convert this into urea in order to make it less toxic. And that's what we do. That's what the liver does. It takes excess ammonia that we use during amino acid catabolism and produce urea. Urea is pretty inert, honestly, in your body, and is going to move freely across cell membranes because most cells have urea transporters. And in elasmobranchs, at least, it is used as what they call an osmolyte. So an osmolyte is when you use some compound, or in this instance an organic compound, as a way to maintain osmolarity. Right? And so elasmobranchs do that, and so do crab-eating frogs. So crab-eating frogs, this little frog that lives in Indonesia, that lives on islands where there's really no fresh water, or very little of it. They feed and lay their eggs, and their tadpoles are raised in saltwater tidal pools. And they're the only amphibian in the world that can survive saltwater. And the way they do it is the same way. They concentrate urea. Right? And the hormone that does that is a hormone we're going to use in lab on our frogs that cause our frogs to gain weight because that causes them to absorb water. These use the same thing. It's called vasopressin. All right, so vasopressin, or what they call ADH, is the main hormone that is going to allow these crab-eating frogs to concentrate urea before they enter the tidal pools. Okay. Yep.
So like that, you said that hormone makes you gain water?
Yeah. Well, it does in us.
Yeah.
But in these guys, it makes them concentrate urea.
Okay.
Yeah.
So if you injected that in me, I would gain water weight?
You could, yeah. It'll make you really thirsty. And you'll start drinking water and produce really concentrated urine. Yeah. Alcohol does the opposite. It prevents vasopressin release, and that's why you break the seal. Well, at least that's what we used to call it back in the day. If you drink too much, right? You got to pee all the time, right? And then it dehydrates you. So. Marine regulators. So marine regulators are most fish, and they are pretty incredible in their physiology. It's a beautiful specific tang here. Can't so you won't. That's over an Indonesian fish. But bony fish are called teleosts. So marine teleosts are the present living bony fish that live in the oceans on reefs and whatnot. They are going to maintain their body fluids about between 300 and 400 milliosmoles, so a little more hypertonic than ours. But salt water is 1,000 milliosmoles. So their body tissues and their interstitial fluid are hypotonic, right? And so they're going to tend to want to lose water or dehydrate because they live in a very hypertonic environment, a dehydrating environment, if you will. And so they're going to have to worry about losing water and gaining salts, right? Because the salts in the environment are going to want to enter into the cells and the interstitial fluid. So they have the exact opposite problem that freshwater fish have, right? So because of that, because they're always going to lose water, they have gills. They're going to lose water, right? They have to drink salt water. I mean, they constantly drink salt water and produce very little urine. All right, the urine they do produce is going to be really high in salt, right, and sodium chloride. They also pump sodium chloride back into the environment, or they convert that salt water into fresh water. And their gills do that. All right, their gills do that again. And the cells here are called chloride cells also, but often they use a different term, which I like better, and they're called mitochondrial cells. And they're called mitochondrial cells because their cytoplasm is packed with mitochondria, which means they have to make a lot of ATP to drive salt out of their bodies. Now, probably some of the coolest fish in the world are those that can move between fresh and salt water. All right, and think about that. To be able to live in salt water and then all of a sudden move into a freshwater environment, that's an incredible feat to be able to do that. You can't take this fish and throw it into a freshwater tank. It dies immediately. And you can't take a freshwater fish and throw it in a marine tank. It dies immediately. But there are fish like salmon, eels, that can move between fresh and salt water. And they're often born in freshwater and then move out to the ocean, spend their life in the ocean, and then come back to freshwater to breed. And when they do that, they are going to basically switch their gills around, you can imagine, because the cells of a freshwater fish are going to do opposite what the cells in a marine fish are going to do, because they're dealing with opposite environments, right? And all you have to do is rearrange your proteins. And so hormones can do that. So hormones can control that, as we'll see. Matter of fact, eels are pretty neat. They're easy to see and understand because they change color as they move from fresh to salt water, and vice versa, because their skin becomes permeable and impermeable to water loss as they move between the environments. So we'll talk about how they do that at the end, but first we're going to talk about the mechanisms that drive this, which will make kidneys easy. Before we do that, let's talk about terrestrial organisms really quickly. So we live in a hypertonic environment. So think about it, right? Air is not water, so it immediately is hypertonic. Even if you're in the jungle and it's 100% humidity, it's still, you're not in water, right? And so your body fluids are, hypotonic to the air. You're always going to be losing water. And so where do we lose water? When we breathe. Our air, and you've been outside when it's cold, you breathe out, it's steam, right? It's because the air that's coming out of your lungs it's been hydrated, right? Or it's got high humidity, if you will. It's got a lot of water vapor in it. And so you're losing water and therefore have to drink water, right? So we have to intake fresh water, at least most of us, mammals do. And our kidneys have the ability now, because we don't have gills, right? I don't have gills. I wish. Our kidneys have to take over that job of concentrating our urine, that is, conserving water. Now, other terrestrial organisms can drink salt water. Now, we can't as mammals. There's not a mammal in the world that can drink salt water and deal with it. But there are animals that can. So there are crocodilians, there are turtles, lizards, and birds, like seagulls, for example. They can drink salt water and convert it to fresh water. How they're going to do that? They're going to have similar cells in a specialized organ as a saltwater fish gill cell that are called salt glands. And so these salt glands, in crocodiles, the salt glands in the tongue or under the tongue. In turtles, it's in the bladder. In lizards, it's in the nasal cavity or near the eyes. Same in birds. And they can remove the sodium chloride and other salts and get rid of them from the body and produce fresh water. Now, the skin of most terrestrial organisms, and this is across all organisms, is really impermeable to water for the most part. The one exception are amphibians. A lot of amphibians had very porous skin. That is, they lose water over their skin. And this is why, where do you find amphibians typically? Are in very moist or very high humidity places, right? Like my frogs, I'm upstairs in my office, they're like, the dart frogs are like this big. So they have really high surface area to volume. And they have evolved to live in jungles with very high humidity. If they get out of their tanks, which happens, I've had a couple get out before, they'll die within like 20 minutes because the water just leaves them and they look like a rawhide in 24 hours. So, and that's sad. I've had one get out before, especially the really little ones, because they're really fast and I open the tank and don't notice, they leave. And then I find them on my desk. It's happened. Yeah, it's sad. So, now there are some amphibians that have very thick skins, like the toads, and there are some desert-dwelling toads that actually live in some of the drier places in the world, but they spend almost their entire life underground because they can't just sit in the sun. They lose water. Some of the most water retentive animals that can resist desiccation, are reptiles. So they do it really well. Now, I just told you that there are no mammals that can drink salt water. So you might wonder, well, how do marine mammals survive, right? If they can't drink salt water. They have really good kidneys and a really good digestive tract. They don't drink salt water, okay? They can't. So but they have to eat, right? And so when they eat, they are going to ingest salt water, right? Whether they want to do it on purpose or not, their gut is really impermeable to salt. All right, and so all the salt that they ingest when they're eating leaves in the feces. They have kidneys that can concentrate water and also kind of get rid of salt really, really, really much better than we can. And they get all of the fresh water they need from what they eat, and they can make water metabolically. So you know that all whales, all cetaceans have really high blubber content. And yes, it helps them maintain their body temperature, but a lot of these guys spend most of their life or part of their life in warm water, right? And they still have high blubber content. And they use that fat to make water. So they burn it. They're constantly burning it to make water. That's why they have to eat a lot to maintain their blubber layer so that they can produce enough fresh water to survive. So they can make metabolic, it's called metabolic water. That's how they do it. You cannot drink salt water. You all know that, right? If you get stranded in the ocean, drink your urine. Don't drink salt water. It'll kill you quickly. You go crazy first. Most people do. Okay, makes sense? The difference between fresh water, salt water, terrestrial environments? Now, because terrestrial organisms lose water to the environment, right? Because we live in air. Let's see if I got a couple. Chalk. I hate chalk. All right. Get a couple colors here. Okay, so because we lose water to air when we breathe out in particular. So here's what's interesting. When you breathe air in, and really dry air, you probably have noticed this, you dry out, right? Winter is a good time when you dry out. Your skin gets dry, your nose gets dry and crusty, right? If you live in a house that has got very dry heat. And the reason for that is because when air comes in that's very dry, you would be surprised. Have you guys all seen the inside of the nasal cavity? Is absolutely humongous. So from here back, like if you're going backwards towards the back of the head, dorsal side of the head, our nasal cavities are quite large. And the reason for that is because they're covered with an epithelium that produces mucus. And we all know mucus is high water content, right? So the air that you bring in is going to swirl around the nasal cavity. There are bones that do this, that kind of meander through the nasal cavity. And when the air comes in, the air swirls so that it makes contact with all of the mucous membrane in there to warm up if it's cold air and to be humidified, to add water content to it. Therefore, the air that then finally enters the lungs is of good temperature and good humidity, right? And it'll make exchange of gases easier in the lungs and feel nice, right? It doesn't, you're not bringing in cold, dry air or, warm, dry air. But when you exhale, you're going to lose all that water, right? That's in the air. And you're going to lose water anyway, but we can minimize that, right? And the way we minimize it is by utilizing a temperature gradient at the surface of the epithelium of the nasal cavity in the nose that is close to the exit point, your nostrils, where you breathe in and out. Okay, and this type of system is called a temporal countercurrent exchange system. Some animals have really good ones, like this cute little dude here. All right, so this cute little rodent is called the kangaroo rat. And it's a small desert rodent, and this is what it does during the day. It sleeps in its burrow, which is much cooler than the air of the desert, right? And they're nocturnal, and they come out at night when the temperatures drop and are more tolerable for a mammal. They have very efficient temporal counter-exchange systems. So the way this works is, what happens to water when it hits something that's cool, all right? When water hits something that's cool and it's warm, the water condenses out of the air and forms water droplets, right? This is what produces rain. This is what produces fog. It's what produces what we call dew point, right? When the water comes out of the air. And so if we have really warm air coming up from the lungs at 37 degrees or 38 degrees Celsius, if we can keep this epithelium cooler than that, right, then some water will condense out of the air and we can reabsorb it. So how do we do that? Well, we already have cool air coming in, like 28 degrees Celsius, right? So we got air coming in that might be much cooler than the air in the lungs. And so if we can figure out how to use that cold air to keep the epithelium cool, then we might be able to reabsorb some of the water before it leaves. And the way we do that is by cooling the blood that is feeding the epithelium. All right, and so the way we do that is through what's something called countercurrent exchange. And so if I just make this the nasal cavity, right? So this is the nasal cavity here. And we have an epithelium, right, that's going to line the whole thing. And so I'll just draw some nuclei in here. And we know this is covered with mucus, right? So the air that's coming in is going to be cool, and it's going to hit that epithelium and cool it down. Now, typically in the body what happens is we have warm blood that has to come down, and we're going to form a capillary bed here, right? Underneath here, and I'll draw little circles to represent a capillary bed. That is obviously going to be feeding this epithelium, like giving it its nutrients and whatnot, right? Now, if we can somehow keep this capillary bed cool, like at the same temperature as the epithelium, then the warm air that's coming out here, right, because it's going to be much warmer, will hit this epithelium, and some of the water will condense out of it, and the epithelium can reabsorb it. But this epithelium has to be much cooler than the air coming in. So the way we do that is we want to prevent this from warming up. And so we arrange our blood vessels that feed this capillary bed so that they come down right next to each other like this, okay? And they give rise, they give rise to this capillary bed here, okay? And so on this side, we'll call this the artery, right, that delivers the warm oxygenated blood, and we'll call this the vein, right, which is the cool blood. So instead of letting the artery and vein kind of run away from each other, we keep them right next to each other. And blood will be moving in opposite directions. This is why it's called countercurrent exchange, okay? Now, we know this cold air coming in is cooling this epithelium, which is likely cooling the capillary bed, correct? But if we allow warm air to come, or warm blood to come in, it'll just warm it back up. So, but the blood that's leaving here is going to be cooler, right? So let's say it's at 28 degrees right here because it's dropped 10 degrees. All right, the blood coming in up here is going to be, 37 degrees Celsius, right? Now, these bloods are next to each other. So this is going to be a cooler supply of blood leaving and warmer blood coming in. And so heat is just going to move over. It's going to move from high to low, right? And so we're going to slowly warm the blood up and as this blood comes down, the heat is going to move across like this. And this is what happens when you have countercurrent exchange. And so your temperature is going to drop, right? Like that. And so that keeps the capillary at the same temperature as the air, and the warmth of our body is going to enter the vein and be swept into our body core. And so right at this, if this is 28 degrees, then right at the epithelium is going to be 28 degrees. The capillary bed is going to stay at 28 degrees because all the blood that's leaving is sucking the heat out of the blood coming into the capillary bed. That's called thermal countercurrent exchange. Have you ever wondered how a duck can run around in the water this time of year and be happy? Right? I mean, you see ice open up and these ducks are just, geese are swimming around, they're eating, they're having fun, like they're swimming on a summer day, because they have the same setup in their legs. The blood that's entering the legs is going to be really warm. The blood going into the body from the legs you would think would be really cold. But they're in a countercurrent setup so that the warm blood going into the legs, the heat is soaked back up so that the blood entering the body is the right temperature. Their legs are cold, but their body isn't. Does that make sense? Deer are the same way. They got those long, skinny legs. How are they not freezing? Because again, they have this same setup. Any blood that enters the legs is going to cool, and then that blood going back into the body should cool the whole body. But if their blood vessels are next to each other, then all the heat is added back to the blood that's going back into the body. Their core temperature stays the same. So that's how countercurrent exchange works. So these guys here are really good at it. So they're absorbing a lot of the water before, they breathe out. So as the air goes out here, water droplets are going to form on the surface of this epithelium, and the epithelium take some of that water back. Now, do they take all of it back? No. They take some of it back, not all of it. But at least you can recover, right, some of that water. So one of the cool things about the kangaroo rat here is because it sleeps and uses the cold burrow, the air in the burrow is already 10 or 15 or sometimes even 20 degrees cooler, because they're like 2 or 3 feet underground, than the 110 degrees in Phoenix, Arizona, where these guys live. And so they can really take advantage of the temporal countercurrent exchange system because the air in their burrow is cooler than their body. If they were trying to do this in 110 degrees, there's no temperature difference, right? So this wouldn't work. And that's why they're nocturnal. We do the same thing. We typically keep our temperatures in our rooms and houses at less than our body temperature. Now, if you're in the desert, this isn't going to work, is it? Another reason why you dehydrate so fast. Now, why did I spend so much time teaching that? Because we're going to use countercurrent exchange in other places. Like the kidneys, they're going to use it. But we're not going to be moving temperature. We're going to be moving salts using the same system. Does that make sense, how it works? Yeah? The gills do it too. All right. So try to draw it. Now, this is probably one of the coolest experiments ever done. So they took kangaroo rats and laboratory rats of the same size, like a normal white rat that you can buy at like the pet store, right? And they put them in the same room with the same temperatures, same relative humidity, which was really low, and then they didn't give them any water. They fed them, they didn't give them any water. And so the kangaroo rats were completely happy. After about a week, the laboratory rats were not happy. Right? They were actually suffering. And the reason for that is because laboratory rats cannot concentrate their urine like these dudes can. All right? So their claim to fame is not just being cute. They are probably one of the best mammals per body weight that can concentrate urine. All right? Better than any other mammal in the world. We can concentrate our urine, you and I, as primates, we can concentrate our urine about 4 times that of blood osmolarity. All right, so think about that. So if your blood osmolarity is 300 milliosmoles, you can produce a concentrated urine of about 1,200 milliosmoles if you're really dehydrated. That's when you produce that really smelly yellow stuff that you're like, Jesus, I need to drink something. I mean, it smells really bad, right? These guys can concentrate their urine 14 times their blood osmolarity. They basically produce paste. I mean, it's not even urine, it's not even wet. There's no water in it. They can also completely dehydrate their feces. So it's like, really dry poop, if you can imagine that. Like you see really dry dog poop, right? So they don't lose very little water from their waste products. Rats, they can't do that, right? And so notice the real big difference. There's not. If you look at these in the beginning here, they lose about the same amount of water over their respiratory epithelium, which makes sense because they have the same kind of surface area. You look at their urinary water loss, though. Look, it's much different. That's very significant physiologically, 0.1% there. And look at the feces. They lose no water, and rats are pretty good, but they still lose water. If you let them do this for a week, they become really dehydrated. Where these guys, look, they actually start making water, right? Because they're making it through their metabolism and not losing any. And that's all it takes, right, is this difference, which seems small, but physiologically it's huge, right? Because any water loss or salt gain or salt loss, if you don't compensate for it, is going to eventually affect you, right? And so they end up having a positive water gain without ever drinking water. As a matter of fact, these guys don't even know how to drink water. They don't drink out of water bottles if you keep them in a laboratory, because they don't drink, they don't see water, standing water. It takes them a long time to learn how to drink water because they don't need to drink water. They just get it from the seeds they eat. They're seed eaters. And then they use fat in the seed to make water. So they're really cool. And we'll talk about, how they do this. So you can imagine what's going to be different is something in the kidney must look different in them than other mammals. Form equals function, right? So we're going to change something a little bit or modify it so that they can concentrate urine. All right.